under human. Yeah, so you're excused yesterday. Are you excused anymore? Those who have not eaten, please just take this time, put something in your mouth that's food related. Okay? I don't know. That was a very weird laughter. Can't tell we came from Keller or Chinese.
Yeah, obviously.
Okay. Okay, can you stand by 1.4b? Sorry, I put a due date as today because I thought we were very behind time. Actually, our class is a bit too ahead of time, so we'll take things slow. Because every time I think there's HPL, I think I'll miss lessons with you. But then on Friday, we actually miss, no, week 10 we are missing Thursday, right? Exactly, so we are not missing anything. So we have 4 full lessons together. Yeah, so it's more than enough for whatever we want. Yeah. Okay, may I get you to go to 1.4b today? Okay, we're going to complete our nucleic acids. For today, at the start, as usual, we'll do a little bit of recap. Who do this job? Huh? Sorry, it's not a dog. It's Meryl. It's Meryl's dog. It's more like a raccoon than a puppy. Okay, so let's do a recap of nucleic acids. After recap, we're just going to do one thing today. We're just going to compare DNA versus RNA. Okay, that's all we're doing today. Okay, first and foremost, may I get you to stand by 1.4b? Okay, the first thing we're going to do today is to recap the structure of the DNA. Okay, before we begin, we just do a quick buzz with your neighbor on the structure of DNA. Then we'll do a quick recap. Okay, ready? Yeah, the structure of DNA. Let's list down as many structural points as we can. Okay, first structural point. Repeat. Okay, it contains 5 carbon sugars. Okay, so at the core of it, when we look at each nucleotide, okay, when we look at the monomer consists of a 5 carbon sugar. If we want to be specific about it, what kind of 5 carbon sugar? Okay, yes, so pentose implies already it's a 5 carbon sugar. Okay, so but there are two kinds of pentose sugars.
Deoxyribose.
Okay, thank you, Jiaqi. So, deoxyribose sugar. Okay, that's one of the distinct things. What defines a deoxyribose sugar? That makes it distinct from a ribose sugar. Okay, if we look at carbon 2, hey, you want to go around for this right? Okay, so carbon 2, and carbon 2, okay, let me draw this thing. Okay, carbon 1, carbon 2, when we look at carbon 2, deoxyribose contains hydrogen bonded to it. But if we look at ribose sugar, at carbon 2, we have OH. So when you see deoxy means it's missing oxygen. Okay, thank you. Thanks, Rin. Jiaqi. Okay, next, any other structural features? I see.
The nitrogenous bases.
Okay, the nitrogenous bases. They also contains bases. We call it nitrogenous bases because it's made of a lot of nitrogen. Okay, Tyson, can you give me examples of bases found in DNA? A, T, G, and C. Okay, could you share with us how these bases interact with each other?
There's like one on each sugar. Then they can hydrogen bond together.
Okay, so they can interact via hydrogen bonding, right? Okay, so the bases can interact via hydrogen bonding. The next level is this. Which bases complement each other? You remember? Oh, shucks. Okay, we wrote it here already. So, this is the pairing you want to look at. A, T, and G, C, that's how they pair with each other. There's a term we use to describe this pairing. We describe this pairing as complementary base pairing. The base refers to the base we're talking about. Complementary means they complement each other. Pairing means to pair. Okay, so complementary base pairing. You can observe that along the two strands. Between G and C, G and C, C and G, A and T, so on and so forth. Okay? Any other characteristics of DNA? Do you know what you are doing here is to describe the characteristic of its monomer, each individual nucleotide. Are there characteristics of the DNA molecule? Sorry? Okay, helical. Kind of like the chapter we just came from polysaccharides, right? Except that what we use here, not so much helical, we use the word helix. It has a helix. Okay, it comes in a helix. And then you often see this word in front also, double helix. Okay, it is a helix. The word double implies that it is double-stranded. Okay. Any other properties other than it having, shapes, helix shape, double-stranded? Any other properties? Yes. Okay. What about the phosphate group? Can I have 1 to 3? What do you mean by 1 to 3? Oh, okay. So again, we look at the monomer, right? The monomer also comes with the last thing, right? Here is bringing up the last part of a nucleotide, the phosphate group. Okay. For DNA, the phosphate group, each nucleotide only comes with 1 times phosphate group. Okay, so just take note of that. I know I gave you examples of nucleotides that can come with 2 or 3. But in the case of DNA, we work with nucleotides with only 1 phosphate group. I will give you examples of nucleotides that come with 2 or 3 later on in the later chapters. Okay, so again, characteristics of the monomer. How are the monomers bonded to each other? What bond do they use to bond to each other, the nucleotides? Okay, there are 2 kinds of bonds. There are bonds that hold the nucleotides together, and then there are bonds that hold the 2 strands together. We need to be able to differentiate the bonds. So what are the bonds being used at each point? Ready? Okay, Jaden? What are the bonds holding the nucleotides together? Sorry? Sorry? Hydrogen bonds. Okay, so holding the nucleotides together, hydrogen bonds. Is it? Okay, so we zoom into what the structure looks like. Where can you find the information you need? If you go into your Google Classroom, open up your notes. Okay, if you were to scroll down to this particular page over here, okay, page 2 of your notes shows you how the nucleotides bond with each other. Okay, and they bond with each other from carbon 3, between carbon 3 and the phosphate group of carbon 5. After bonding with each other, this is what you observe. And we describe this bond to be a phosphodiester bond. Okay? What Jaden brought up about hydrogen bonding is present. Where do we find that? Okay? Jaden, where will we find the hydrogen bonds? Yeah? Sorry? Between the two strands. Okay? You can expect to find the hydrogen bonds between the two strands. Specifically between the bases. So this is where I will label the hydrogen bonds. Hydrogen bonds. Right? Hydrogen bonds, phosphodiester bonds, link the nucleotides together. Hydrogen bonds. What's the word to use? Hydrogens connect. It's not connect. It's not condense either. Okay. The two strands interact via hydrogen bonds. Okay, so just make this distinction. I find that this is the one point that usually students will swap over. There's one more characteristic of DNA, just one more. It has to do with the idea of a 3' end and a 5' end. Okay, yeah? It is anti-parallel. What does that mean? First and foremost, if you were to just work with your partner, can you identify the 5' end and 3' end of the top strand, the 5' end and the 3' end of the bottom strand? Okay? Let's do a quick exercise.
Wait, so you said just now it has two types of structure. One is the double helix and one is the double stranded.
Oh, double helix describes the twist that you observe.
Twisted ladder.
Yeah, yeah. But double stranded is just simply that it consists of two strands.
So double stranded just means two strands, but double helix refers to the shape of it.
Yeah, that twisted ladder shape.
Wait, so it's always double stranded?
Yes, DNA is always double stranded. Okay. Sorry, okay. Have you identified the 5' end and 3' end? Okay. Okay. How do you tell what's the 5' end, what's the 3' end? Yeah. To understand what's the 5' end and 3' end, you want to again go back to this structure here. 5' end will be the end that has a free phosphate group. Okay, at carbon 5. 3' end will be the carbon that has a free OH group. Okay, so how will we identify the groups over here? Okay, Tarunika, the top strand. That side is the 5'. This side is the 3'. Okay, and we go to the bottom, 5 and 3. So the idea that each strand runs anti-parallel to each other, that's what we will use. Anti-parallel. Okay, that they run parallel to each other, but it's anti. Right? One goes from 5 to 3, one goes from 3 to 5. Okay, that's what it means. These are the general structures of a DNA. What I've just shown you on the whiteboard is a very short segment of DNA. Not even twisted in any way. Real DNA strand will be really, really, really long. Hundreds and thousands of bases. I thought I lost my DNA molecule. Turns out it's in O3 sitting somewhere. Like running around a staff room, cannot find it. Very expensive. Okay. So that was a recap for today. I'd like to take any questions at this point from anyone on the ground, on the floor. Yes?
The anti-parallel thing.
The idea of anti-parallel is that one strand runs from 5' to 3' end. The other strand goes in a 3 to 5 direction.
So that's all.
Yeah, that's all it means. Yes? So then you may wonder, right? I mean, I ask myself this question. Why must it run anti-parallel? Why can't it just go in the same direction? 5 to 3, 5 to 3. Yes?
I don't really understand like the 5' and the 3'.
Okay. Are you able to identify a 5' end and a 3' end first? Okay, so we go back to again the structure of a nucleotide. Yeah? Okay, let's look at the structure of a nucleotide. Here's a typical structure of a nucleotide. Can you see that the carbons are numbered? 1 to 5. Yeah. Can you see carbon number 3 and carbon number 5? Carbon number 5 is where the phosphate group is attached. Carbon number 3 is where, if I scroll down, okay, carbon number 3 is where, let's look at this nucleotide here. Carbon number 3 is where the OH is attached. Okay? Every time we want to elongate a DNA, we want to add more nucleotides onto it. Right? Notice that to add a new nucleotide, we will take the phosphate group bonded to carbon 5 to bond and condense with carbon number 3's OH group. As a result, we will get a phosphodiester bond. Right? Okay, so this is still okay. Now if I were to zoom out, the reality is that every time I add a new nucleotide, there will still be one end of carbon 3 that has no nucleotide bonded to it. Yeah? So we call that the 3' end. Carbon 3, why is it called prime? I don't know why it's called prime. Yeah, think of it as a free end. A 3 free end. Okay, but it's a 3' end, that's how we describe it. The other end is the 5' end. That's the end that does not have a nucleotide bonded to the phosphate group also. Okay, so that's how we describe a 3', 5'. Okay, so back to my question. Yes? Yes? Ribose. Okay, DNA will always make use of this particular sugar. That's why we call it deoxyribonucleic acid. That's the full name of DNA. Okay, so if you ever forget whether it is deoxyribose sugar or ribose sugar, the name DNA actually stands for deoxyribonucleic acid. Okay. Yes?
So the ribose one is only for RNA?
Right. Okay, so RNA stands for ribonucleic acid. So in a while, we will look at RNA. Right? RNA makes use of the nucleotide with this particular sugar. Good. Any more questions? Okay, so my question was, why not the strands go from 5 to 3 and 5 to 3? Why can't they parallel? Yes? Okay, the bases must face each other. If I were to try to do this 5 to 3, right, both of them 5 to 3, you find that the bases no longer complement each other. Possible? Okay, that's possible. But this is the configuration we already have. Could I have complementary base pairing but still be 5 to 3? What if I start off with complementary base pairing, but they run in a 5 to 3 direction? The best I could find online about why things don't go parallel to each other is that when they go parallel to each other, the way the bases are arranged at the microscopic level, the hydrogen bonding is not as ideal and not as comprehensive. So it is possible, but it is just not as stable as an anti-parallel configuration. For your information, okay? In case you're like, why? Okay, so take some time to touch up your notes, because the next part of today's lesson is what we will do. I'd like you to go to 1.4b In 1.4b, we're going to compare the structure of DNA and RNA. Just now 5 people submitted, now 6. Who's that 6th person that suddenly did it and submitted? Funny, huh? Okay. I mean, it's good. Okay, so what we're going to do now is this. Number 1 is for your own doing. You can do it at home in your free time. If you want to learn a little bit more about who discovered DNA. You watched it? You did it, okay. It is. Number 2, okay, is what I would like us to do today. I'd like us to quickly, okay, I'm going to screen this on the screen so we watch together. We're going to watch around 5 minutes of video describing the structure of RNA. Our job is to compare the structure of DNA with the structure of RNA in the video that we're about to watch. Okay, that's the skill to pick up today, and that's all we're going to do today. Should I leave the background music?
Yeah.
Okay, are you ready? Okay, the task now is to compare RNA structure to DNA structure. This video is rather in-depth. If you go to this website called iBiology, they actually managed to get a lot of the original scientists that discovered their own, that made the discovery to teach whatever they are teaching. I'm not sure if this lady was the one that, I don't think so, but I've seen the actual scientists teaching their own work. Some of them are really, really old. Let's see if you can hear her. Hey, why is she not coming?
You have to stop the jazz.
I see.
No.
How can you use this?
From Yale University, and today I'd like to tell you about RNA structure. And I'm hoping by the end of this seminar that you'll be thinking about RNA as a much more complex molecule than you might have thought about it before. One of the things that's important to keep in mind is that for any biomolecule, very often one sequence will lead to a unique structure. And this is true for proteins, and it's also true for many RNA molecules. And today I hope to tell you how that can be achieved. Now, some RNA molecules are actually unstructured, like messenger RNAs that contain the code for translating a protein. But other RNA molecules have elaborate structures, such as tRNA molecules, ribozymes, which are catalytic RNAs, the ribosome, which is the factory that makes your proteins, and riboswitches. I'm showing you a riboswitch in this image here. RNA molecules are often highly complex in structure, and it's important to remember that structured RNAs are involved in almost every aspect of gene expression. I'm showing you some examples here. In this case, this is one of nature's biosensors. It's called a riboswitch, and it's important for bacteria so that they can sense metabolites in their environments and signal back to the cell. This is a catalytic RNA called self-splicing group 2 intron, and it's a molecule that can cut and stitch RNA and DNA molecules so that they can go on to be functional. And here, this giant RNA is your protein synthesis factory, which ironically is an RNA molecule. And it is probably one of the largest and most highly structured RNAs in our cells. But even if you're most interested in the function of messenger RNAs, it's important to remember that many messenger RNAs also contain highly structured regions. So even though the middle part of an mRNA molecule often just contains the coding region, the triplets that encode the amino acids for your proteins, often the RNA sequences at their termini form these complex structures in the untranslated regions, and these help to regulate the process of protein synthesis. So the chemical structure of RNA is a big part of how it can achieve these many functions and how it can adopt these complex structures. And a couple of very important things distinguish RNA from its cousin, the DNA molecule. So in addition to the 2' hydroxyl groups that you see here, the uracil base looks a lot like thymidine, but it's missing the 5-methyl group at this position. So one of the bases on RNA is different than it is on DNA. But back to the 2' hydroxyl group, which is really the thing that endows RNA with special capabilities. I like to say the 2' hydroxyl group gives RNA special powers. And the reason for that is that this substituent can both accept and donate a hydrogen bond. So every 2' hydroxyl group can make up to 2 hydrogen bonds. So the bottom line is that makes the RNA backbone very sticky and enables RNAs to make a number of types of interactions that are impossible if you just have a deoxyribose at this position. In addition to the chemical structure of RNA, another thing that gives it its shape and distinguishes it from DNA is the structure of the sugar moiety. So a lot of times people think that the things that make DNA and RNA molecules special are the sequences of the bases and the base pairings between them. But a lot is going on with the sugar phosphate backbone. And in particular, it's important to remember that sugars are not flat. Because they're 5-membered rings, they often buckle or pucker. And this gives both RNA and DNA their distinctive shapes, and we'll be talking about those more in a moment. There are 2 major conformations of the sugar pucker in a ribose, and one of them is the C3' endo sugar pucker. And so if we imagine the sugar is in this position here, the 3' atom of the sugar pushes up. I like to say it's pushing in toward the plane of the base, and this gives you the C3' endo conformation. The consequence of that conformation is that the interphosphate distance is very small. It's only 5.9 angstroms in an RNA-like sugar. However, these sugars are always flexing, especially if they're in the absence of surrounding structure. And an alternative conformation that can be adopted by ribose is the C2' endo conformation, which is actually the one that's dominant in DNA. And that occurs when this carbon kicks its knee up and the C2' position becomes closer to the base. The consequence of that is that the interphosphate distance gets very large. It's 7 angstroms. And that's why you'll see later in the presentation that DNA duplexes are more elongated and spread out than RNA duplexes. It also is an important factor in giving RNA and DNA their helical shape. Okay, so now that we've covered some of the most important aspects of primary structure in nucleic acids, let's talk a little bit about the secondary structure. And some terminology is helpful. First of all, it's important to define RNA secondary structure as the regions of RNA base pairing. So I'm exemplifying this with the second.
Okay, so we'll pause here, right? Quick parts. What differences do you take note of between RNA and DNA? But it's extremely high level. Okay, so you see what you got, then after that I'll show you a simpler. Okay, ready? Alright, okay, this video, right, is a Yale University level video. Okay, but what do you manage to tease out? Okay, for the parts that you understood? DNA versus RNA. Caleb, what do you tease out?
RNA, they use uracil and DNA use thymine.
Okay, RNA, rather than thymine, they use uracil instead. Okay, so in RNA, the bases involved are A, U, C, and G. These two complementary base pair to each other, these two also. But in DNA, it is A, T, G, and C. Okay, if we were to spell out U, it's uracil. Oh, sorry. No particular order. You can written it as CG or GC. Yeah? I just put the two of them side by side to show them that this is a pairing. Okay, this is the complementary base pair we're looking at. Thank you, Caleb. Okay, so that's the first difference. Difference number 2. Isabel? Okay, so if we look at the carbon number 2, okay, at carbon number 2, you find that for RNA, you find a hydroxyl group. Okay, again, I want to emphasize this is called hydroxyl group. It is not a hydroxide group. Okay, it's a hydroxyl group. Okay, and in the case of DNA, when you look at the sugar, carbon number 2, it bonds with a hydrogen instead. Okay, so that's one of the differences. Any other differences? Yes, Christine? Okay, so now let's look at the structure. For DNA, it has a structure. It is this double helix structure. But when we look at RNA, it can be unstructured or structured. Any more differences? Yeah, so just let you know, just now that there's one part where they say the sugar cockles and kicks up the knee or whatever. Okay, that one is university level stuff. Okay. But I let the video run up to there. Okay, because we are O2, you're supposed to like science. So let it screen a little bit more. Okay, any more differences? Valentina? Any more differences? It's okay, go ahead. No? I've not heard from Santosh. Any more differences?
They say about the C3 endo.
Okay. Okay, so Santosh, I think like many of us heard this C3 endo thingy. Okay, so we look at that. Let's go back here. This one over here. Okay, so it's out of our syllabus. Just to help you, let you appreciate this a little bit more. Every time we draw on the whiteboard nucleotide, actually we have flattened everything out onto 2D plane. In reality, nucleotide is not flat. If we take reference to the base as a flat one, the sugar actually pops out at a different plane towards you. Okay? The phosphate group also is on a different plane. So when you start building your DNA molecule, when you start your competition, you realize that actually it's not possible to form the double helix if all the sugar, the base, and the phosphate are on the same flat plane. Actually, they are on different axes. Okay. So what does 3' endo mean and 2' endo mean? What she was saying is that 3' endo means that actually for RNA, the sugar, and carbon number 3 actually pops upwards. Carbon number 2, it pops, for RNA, carbon number 2 pops upwards. Okay? This one would help you understand and appreciate why RNA looks a little bit more compact and DNA is a little bit more spread out. Okay? Okay, but so I'll let you know now, this one is beyond our syllabus. Okay? So this was for your information. Last difference, let's look at the structure of RNA a little bit up close. Let's, for example, look at, it looks like a mess, right? Okay? Sometimes it looks structured, sometimes it looks unstructured. One of the key things about RNA is this, compared to DNA. Let's look at this particular RNA over here. Okay? This is actually quite a structured RNA. But if you look at this RNA, you know this is just one strand. Yeah? RNA is single-stranded. Single-stranded. DNA is double-stranded. Yeah. Okay. However, because of the sticky nature of the OH group, it allows the RNA to bend and fold upon itself to form these structures. So even though it looks like, it almost looks like it's double-stranded, but actually it's folding upon itself. That's something RNA is capable of doing. Lots of hydrogen bonding taking place. Sorry? Sorry? Oh, this is the side view. Okay, again we flatten everything out. Yes?
Wait, so you're saying because of that hydroxyl group, it makes hydrogen bonds within itself?
Correct. Yeah. So it is, if you want to use a more chim word, it means intramolecular hydrogen bonds. Okay, intra means within, molecular means molecule. So these hydrogen bonds are within itself. So actually for DNA, it is intermolecular hydrogen bonds. Yeah, I don't expect you to use this vocabulary because actually I find there's an easier way to describe this. But if you want to, you could use this vocabulary. Yeah? Okay, these are the key differences between DNA and RNA. I'm going to teach you a trick when it comes to answering questions. When it comes to worksheets or exams or quizzes, if we ever ask you to compare and contrast two things, you can actually draw a table and submit them. Okay? Yeah? Even A-level side accept it. You can draw a table to make it very clear your comparison. However, if you want to draw a table, you would need to have one more column for the characteristic you are comparing. Okay, just to make it very clear. So for example, here I will write maybe something like, yes, bases. Maybe over here, type of sugar maybe. Okay, type of pentose sugar. Okay, this one after all is a deoxyribose. This one is a ribose. Here you can put maybe the structure. Last one, perhaps number of strands. Okay, you can submit a table. Okay, but here's a, I don't know how to pronounce this, that you do have to write it out in prose. So what I've done over here is short form. Yeah, just put the vocab in. You still need to write out a sentence of some sort. So for example, DNA has a double helix structure. then okay, so do write it out in sentences, just so that we can really understand what you're writing here. Because if you were to just write this, that I've just put up here, 2C then H, 2COH, makes no sense. Okay, so you write it out in sentence. Alternatively, if you do not want to draw a table, it's fine for you to still compare the points. Now, I want to say something as I'm marking your carbohydrates TE. I noticed that many of us like to compare paragraph by paragraph. What do I mean by that? They will lump all the characteristics related to DNA in one paragraph, and then all the characteristics of RNA in another paragraph. Okay? Instead, if you do this, could you compare point 1, DNA versus RNA? Maybe the first point is on bases. Then point 2, second paragraph, I want to compare type of sugar. Then maybe paragraph 3, I want to compare the structure. Okay? Then you may ask, actually, what's wrong with doing this? Okay, the only danger of doing this, right, is that sometimes students may list out 3 points here, and then here they list out 2 points. Then I don't exactly know what you are comparing. Okay? Because I would assume that, oh, you're comparing the first point over here to this first point, second one to this, then third one compare to what? Okay, so that's the first danger. Second one is that sometimes you don't order the comparison in order. Maybe this is point number 1, 2, 3, but over here you write the third comparison point here, then you write the first comparison point here. So I don't know what you're comparing when you put in paragraphs. So instead, do it this way. Then the last reason why it's not so good to do this way is because imagine eventually when you go to A-levels, you submit the script all the way overseas, you're marking thousands of scripts, right? They may miss out your point of comparison. Okay? So if you do it point by point, it's easier to spot all your comparisons. Alternatively, you can do a table. There's no way to miss. Yes?
So, you know, when you said about doing a table, you know, the question will ask, and the question will give you all those lines. So, using those lines, you just draw a table.
Yeah. So, what I see students do is that all the lines there, right? They draw one line in the middle, another. They basically drawing vertical lines so that they can write.
Like, usually you draw the horizontal one, you just draw the vertical.
So, I see students do that. Some students use the space below to draw a table. Closed table. A closed table. Okay? Okay. So I put you through all this game. Actually, if you go to Lumos, again, there are a lot of summary videos over there. There's actually just one really short.
Thanks for stopping by. Let's talk about the differences between DNA and RNA.
Okay, this one is really short, quick summary.
So DNA is usually double-stranded. It has 2 separate strands that are connected, which is like this. While RNA is single-stranded most of the time, looking like this. DNA is much larger than RNA, with usually millions of nucleotides. And RNA is, of course, smaller, with hundreds to thousands of nucleotides.
It's something we didn't discuss. DNA is larger than RNA.
DNA has a deoxyribose sugar, that's why it's called deoxyribonucleic acid. Its sugar structure looks like this. And the DNA, of course, has the phosphate group, missing the carbon here.
So by the way, if you want to create educational videos next time, let's say, I don't know which subject asks you to create a video, you could go to Video Scribe, then you can input and get them to animate this for you. See this drawing, it's very nice, right?
The carbon has an
they draw this, actually it's all a function you can find in the Video Scribe.
The location within the cell for DNA is primarily located in the nucleus, with some DNA being in the mitochondria.
This is another difference that I don't think we discussed or was mentioned in the video.
Or on the right.
Okay.
While some
DNA is primarily found in the nucleus. That's what we already know. Okay, where do you find RNA? Actually outside the nucleus typically. We find a lot of it floating around in the cytoplasm. But there's a lot of spelling errors. This one is nucleus. You ever watch Blue's Clues when you're much younger? I love Blue's Clues. You know a movie for Blue's Clues is coming out? when they bring back all three Blue's Clues men to the movie. I don't know which generation of Blue's Clues you watch. I watched the one with, what's his name, Steve? I never watched Bananas in Pajamas. Okay. Could be what? Could be food.
Have you watched Bananas in?
Have you ever watched
What is that?
oh, I love this show, it's called Bear in the Big Blue House. I don't know if you watch, but then watch before, it's very nice. Oh my. Teletubbies, right? It's like a little bit off. I won't let my kids watch. That one's a bit off. Okay? Okay, so that's all for today. Yes. Okay, we still have 10 minutes. Okay, let's play Kahoot.
Yes.
Okay, so I've compiled a series of questions of common misunderstanding from lipids, carbo to nucleic acids. Alright? So these are the most common things I see.
You want to put emoji? I think it's not enough.
Okay, the price will be leftover chocolate lava cake kisses.
Do you have the other flavor?
The hearts were all taken away. Yeah. What's the answer today? What's your answer? You get chocolate today. This chocolate is good. A certain circle. You lost the chocolate? But you don't like chocolate. Your potential. Moral cat. Meow, I mean, is that you? Oh, meow. You just came up with it becomes meow.
Not fun.
Okay, 27 of us. Yeah, I know I see this. Okay, I see this over here, but I just want to say what's happening now is not really a laughing matter. Okay, so if you let's stop this movie. I think it will be good if you have not read up on the news to read up a little bit more, okay, so that we can be sensitive about what we say. I think while the impact we may not feel as great an impact, it may trickle down to us. We never know. But I think yeah, we'll be sensitive in this time. Okay. So I cancel this up. Okay, and then I encourage you to read up a little bit more. Okay, then I'm gonna hope that if whoever it is, you can key in again. We still have 27, so maybe that person will be biology. Okay? Yeah? Okay. I know finish. Okay. Okay, ready? Okay, so these are common misconceptions. Sorry, this question stumped me also. When I saw this, I was like, wait, where is it wrong? Okay, you know why it's wrong? Okay, so this is the general structure. No, this is for glucose, C6H12O6. The general structure is not C6H12O6. It is CH2O, and then you see this N. This N means that you can have a variable number of these sets of things. Okay? You remember my trick? You remember no? Wait, were you the class that was unimpressed?
Yes.
Okay. And then after that, you all made impressed on me, right?
No, it's sigma.
Starch. I'd rather know. Hey, oh my, okay. So I guess you're okay. So maybe now, right, you see the use of it. Okay, so Milo bacteria. How you pronounce it? Amilo? Wow. Amilo. Okay, actually that makes sense. Why did I call it amilo?
Alpha.
Yes. We are reaching our full potential. Sorry. Hey, I'm ready for you. Okay. So, nitrogen. Oh my god. Hey, Sharks, can you stop the recording?